Good morning, Hillary and Allie. This is Brian. This is a video lecture that was supposed to be for Friday, February 8th. And I'm starting with protein verification. And this is after purification, but I did want to just remind you of the different things that we've talked about in downstream processing since we started downstream processing. First, after the protein is produced, we had the first step, which was to remove the extract or to get the protein containing phase, either intracellular or extracellular. So we called that first step extraction. Then after we talked about extraction, then we talked about stabilizing the protein extract. So we would add some type of salts and buffers and antimicrobials to stabilize the protein extract. That's step number 2. Then step number 3 was to get the protein fraction or further fractionate the protein, and that was usually via precipitation. And we used ammonium sulfate to precipitate out specific protein products, or you could use varieties of solvents like isopropyl alcohol or ethanol. So the first 3 steps were extraction, stabilization, and then protein isolation. Then we talked about protein purification, and after you isolate the protein fraction, then you go forward to the 4th step, which is protein purification. And in protein purification, we talked about 5 different columns. Okay, the first column was the size exclusion chromatography column that would separate out proteins based on their molecular weight. The second was the ion exchange column, where we would look at the ionic charge. Proteins typically have a negative charge associated with their surface. So we would look at the negative charge versus the positive charge of the ion exchange beads and use that for chromatography, and then use an increased salt concentration to elute out different protein fractions based on the surface charge. So that's the second one, was ion exchange chromatography. The third type of chromatography was affinity chromatography, where we would take very specific affinity ligands that would be specific for the molecule. Say you would have a ligand that would only bind insulin or human growth hormone, and we take those ligands and we would attach them directly to the purification beads. And then under certain conditions, you could elute out only the protein of interest. So the third was affinity chromatography. The fourth was hydrophobic interaction chromatography, which is quite, when you're getting down to a relatively pure product, you want to use affinity chromatography and hydrophobic interaction chromatography. Hydrophobic interaction chromatography was based on the hydrophobicity and the attraction of hydrophobic amino acids on the surface of proteins to hydrophobic beads. And here you would use a decreasing salt concentration in order to elute out different proteins. Finally, there was isoelectric focusing. Isoelectric focusing, you would control the pH of a column system and equilibrate the pH with the PI, the isoelectric point of the protein, and use that for the principle of purification. Okay, so the 5 again, different types of purification methods are size exclusion chromatography, ion exchange chromatography, affinity chromatography, hydrophobic interaction chromatography, and isoelectric focusing. Okay, so then once we have a pure fraction, and like I drew on the board, you're going to have multiple purification steps. First, to get rid of the noise, just to get rid of a lot of the proteins that aren't the right molecular weight, and then finally to purify out only the protein of interest. Once that protein is purified, then it has to be verified. We have to verify that not only do we have the correct protein, but the protein also has the correct activity. So let's go ahead and start with that. So verification is a very important step. And you have to verify that the target protein was not lost during protein purification. And that sounds quite odd, but there are purification schemes where they've lost a protein of interest. because of just faulty recovery of the different protein fractions. So you have to verify that the protein is in every purification step, and then that the protein end product is sufficiently pure, and it's also identical to the protein that they want to produce, and it also has identical activity. The first thing that you want to do is just do SDS page, and you look at all the different protein fractions after all of the different purification steps, and then you can identify the protein. We use a detergent called SDS. It's added to the protein mixture, and the mixture is heated, and when that happens, the proteins denature, and they more exactly will separate out on the gel based on molecular size. And this is because of sulfate charges that are attracted to different amino acids on the protein. These are evenly distributed along the denatured protein, and the denatured protein is linear. So these sulfate charges make the charge potential, which is the principle for separating out the proteins, directly correlate to the molecular weight of the protein. Okay, and here's SDS page, and you can see moving along in this system, your first purification step, where's my arrow? First purification step, you have lots of different proteins. Second, you get less. Third, you get less. Fourth, you're getting almost no protein except for the desired protein. And fifth, as you move along from 1 to 5, you actually see the band for the desired protein actually strengthen and become more intense. And that means that not only you're purifying the protein, but you're enriching the protein fraction. Okay, so lane 5 represents, oh, I'm sorry, lane 5 is a protein marker. Oh, whoops, my bad. Okay, so lane 4, you're getting rid of a lot of the gunk, but you still have more purification to do, and 4 is the most purified of these different steps. Okay. Also, you need to run a Western. You cannot identify a protein just based on molecular size. You have to probe it with an antibody that is specific for that protein. So you run a Western blot. So the proteins are transferred from SDS page to a membrane. All sites on the membrane are blocked, so the antibody won't bind them non-specifically. And then you add a primary antibody, which is specific for the protein that you're trying to purify, and you incubate that with the membrane. And then you have what's called a detection antibody. That detection antibody we call it is a secondary antibody coupled to a reporter group. Okay, the secondary antibody is specific for other antibodies. Okay, it's called an anti-immunoglobulin, and so it will see the primary antibody bind to the primary antibody, and it will have a little reporter group called alkaline phosphatase, and alkaline phosphatase will cause the band to develop, so you can see it. Excess secondary antibody is washed off the membrane, and then you add some type of substrate that binds the alkaline phosphatase, and that results in a visible band. So it's kind of complicated. A Western blot isn't just adding a probe to find the protein. You have to start out with a primary antibody that finds a protein of interest, a secondary antibody that binds the primary antibody, alkaline phosphatase, which is conjugated with the secondary antibody, and then a substrate that will actually become visible when it reacts with alkaline phosphatase. So it's a multi-step process, and that makes it more difficult to run. So this is something that I got from plant-based production, and you can see they're looking at different fractions, and they find that, yeah, they get a weak fraction when they look at the total leaf. If they go down further and look at just the chloroplast, you can start to see the band. They look at the thylakoids and the pull-up chloroplast. The band's still there, but a little faint. And then if they purify out photosystem 1, which is the protein complex that is one of the protein complexes responsible for photosynthesis, then they see their target gene or target protein and are able to purify it and get a relatively pure fraction. Okay, instead of a Western blot, you can do ELISA. ELISA gives you the identity of the protein that you're looking for, but it does not tell you anything about the relative purity or the molecular size of the protein. Let me say that again. ELISA will tell you, it will detect and give you the presence of the protein, but it does not give you any features regarding the purity of the protein in the fraction or the molecular size. Okay, so you get limited information. If you run an ELISA, you will have to run a Western blot as well to verify size and purity. Okay, so but you can define and detect specific proteins. You need a detection antibody and a reporter antibody. And you take this on a multi-well plate, like a 96 well plate, and it uses the principle of affinity chromatography. Okay, antibody 1 is added to capture the protein that you're looking for. Then the protein, the protein sample is then added after antibody 1, and then you wash it and you block excess antibody. Then antibody 2 is attached, antibody 2 is attached to an enzyme, and that enzyme is added, and antibody 2 binds antibody 1, and the substrate to the enzyme is then added, and that gives you a color. Usually the color is green or red if you detect the presence of the protein. So therefore, you get a color reaction if you have the protein in your multi-well plate, and then you can use something like a spectrophotometer and look at the intensity of that color to see exactly how much protein is in that sample. Okay, and here's a multi-well plate that's green. And you can see there's a lot of wells that don't have any green in them. So here probably at the top we have just a calibration curve where you start with a low concentration of protein, get a little higher, 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 and higher. And then you run samples down here. This is a control well here. And you can see in specific samples, and a lot of times these are serial dilution samples. So you start here and you dilute down a little further, dilute down a little further, dilute down a little further until the antibody is gone. And you can see in the second row, you have quite high intensity even when you dilute down further. So you'd probably use this particular cell line. Say if this is E. coli that was producing insulin, you'd probably want to use this particular cell line because ELISA is telling you that you have a lot of protein. Okay, then you also have to preserve the protein. You want to make sure, remember, proteins are WIMPs. They are not very stable at all. So once the protein of interest is isolated, collected, and purified, then you have to preserve its activity. You have to put it in a buffer that mimics its normal conditions, whether it's intracellular or extracellular, so the protein won't fall apart. One easy way to do this is to freeze dry the protein. Some proteins do not survive freeze drying, they have to stay in solution. Generally, lyophilization maintains protein structure. And a lot of different types of proteins you can just freeze dry, and you can even send them to the patient in a vial freeze dried, and they just rehydrate with some type of solution, and they can be stored at room temperature for long time periods. Okay. In terms of scaling up protein production, or purification, excuse me, a lot of these methods, especially the column methods, are worked out on the bench scale, but they may not be adaptable to large-scale protein production. Remember, I showed you pictures of columns that were literally over a foot in diameter. So what may work in the laboratory with a tiny column that has a diameter of less than an inch may not be appropriate for full-scale protein production. Okay, and also you have to be very, very careful with your purification process because that can invalidate your small-scale clinical studies. Okay, so when you have a protein and you're getting it approved for use and as a pharmaceutical, you have to go to the FDA. And so you give the FDA product based on small-scale clinical studies, and they study them in the clinic. However, if your scale up of your purification alters the product, then that can nullify the approval that you get from the FDA to move forward in produce and market your protein. So just small changes in your purification process when you scale up can invalidate your clinical trials and can result in getting the approval for your particular pharmaceutical rescinded. Okay. So why would it be necessary to seek FDA approval for a bioengineered protein that's already been approved if the process is changed or scaled up? And this is where you have to remember, if the process has changed, then there is a possibility that you've altered the protein structure. So for every process change that happens, then you have to get at least a small scale clinical trial to validate that your protein product has not changed. You have to verify that you have the same protein product, even if you change the production process, because again, proteins are very fragile, and just minor alterations in the production processes may mean that you have to go back to the FDA and get approval for your product. Okay. After you purify other analysis methods, you may do what's called N-terminal or C-terminal sequencing, where you just chemically determine the protein's amino acid sequence. You start out with methionine and then go to the second amino acid. That's called N-terminal sequencing, and then the third and fourth, and that gives you the identity of the protein structure. Okay, you can do this very easily now with what's called mass spectrometers. And they will take proteases, and the proteases will chew up your protein into smaller parts, and then they can identify the peptide masses, and based on these peptide masses, determine the amino acid sequence. Another method is to look at your protein structure. And in order to define protein structures, you first have to be able to crystallize your protein. So their proteins will form crystals relatively easily when you bring them out of a solution or a buffer. And so you crystallize your protein, then you shoot X-rays into the crystal based on X-ray crystallography. And based on the diffraction pattern of the X-rays as it moves through the protein crystal, then that will give you inferences based on the protein structure. So if your protein, say it's something that has a complex tertiary structure like an immunoglobulin, or a quaternary structure like hemoglobin that has multiple subunits, then you can use X-ray crystallography, but you do have to have the ability to crystallize your protein before you shoot X-rays at it in the X-ray crystallography and determine the structure. Okay, so you need pure crystal proteins. You just dehydrate the solutions and the crystals will form. And then when you expose the crystals to X-ray, a pure protein creates a specific shadow pattern based on the diffraction of the X-rays and the configuration of the protein. Okay, and so here is the sequence of insulin. Insulin has an A chain, which is shorter, okay, and that gives you the amino acid sequence for the A chain of insulin. Here's the B chain of insulin, which is a little longer. And then you see cysteine residues here, where it says cis and cis. Those cysteine residues form a disulfide bond here and a disulfide bond here, so that covalently holds the A chain and the B chain of insulin together. However, you see it's a really complex protein. Even though it's a small protein, it has had to cleave at the right position. It has to cleave right here. And then after it cleaves, then it has to form disulfide bonds. Ooh, I missed one. Here's a disulfide bond here that is just for the A chain, but without this disulfide bond, you'd get improper folding of the protein here. Here is a ribbon diagram based on the X-ray crystallograph of insulin. And this ribbon diagram shows that you have a bit of an alpha helix here on the A chain, another alpha helix here, and kind of an amorphous structure here. And then you would use this information, use this image, compare it to normal human insulin in your protein production process to make sure that you were actually producing human insulin. So why do you think you need crystallography if you're going to sequence the protein, you already know what amino acids are there. Why would we need crystallography? Well, with crystallography, you get the actual structure of the protein. The amino acid sequence is just one facet of the protein. In order to get the secondary structure, the tertiary structure, and the quaternary structure of the protein, you actually have to be able to image it. And to image it, you use X-ray crystallography. Okay, so you need both. You need to be able to sequence the protein, and then you need an X-ray crystallograph of the protein to make sure that indeed it has the right sequence and the right structure. I'm going to talk briefly about proteomics. I want to check time here really quick, see how we're doing so far. I've only been talking for 20 minutes, and I've just got a few more slides. Okay, so with proteomics, and with the advent of genomics, now everything is omics. You have proteomics, you have metabolomics, you have transcriptomics, you have glycomics. But proteomics is just the study of all the different proteins in a cell at a given time and a given condition for that particular cell. So proteomics is a new scientific discipline, and we're looking at different disease states, and during those disease states, which proteins are being expressed and which proteins are being suppressed. Okay, so you do a comparison. You look at the proteomes, you actually purify out all the proteins between healthy and disease states, and then you look at the difference. You actually subtract out the difference in expression level between the healthy person and the diseased person, and then you can correlate that with the progression of the disease and understand the disease better. So our main goal here is to find protein markers that are either significantly upregulated or significantly downregulated, and you can use that as a diagnostic system to look at the progression of certain types of cancers, of HIV, of autoimmune disorders, and then use those markers as the basis for the development of drugs. Okay, so with proteomics, you can use a gel. Only in this instance, you use 2D gel electrophoresis. Okay, and with 2D gel electrophoresis, you might on one dimension separate out the protein based on size, and then on the other dimension, you may separate out the protein based on some type of charge characteristic. Okay, and then once you do 2D gel electrophoresis, instead of getting bands, you get little dots. You get little dots that you can pull out. You cut out the dots, and then you run them on mass spectrometry, and that will verify. The mass spectrometry will lyse the protein up into fractions and then give you the actual identity of the protein. Okay, so and then when we're done with all that, then we can use amino acid sequencing to characterize a particular protein. Two-dimensional gels are a real pain to do. They're quite large. They're not your typical gel box apparatus. And to run on 2 dimensions and to get distinct protein bands can be very difficult. The other way is to do a protein microarray, and this works a lot like a gene chip. So you just take a set of proteins and you immobilize them on a surface, and then you add a reagent that will indicate binding of the protein by color change. Okay, and in doing this, you're not necessarily just looking at the proteins that are there, but you're looking at the protein, the way the protein interacts with different ligands. Okay, protein interactions in disease and in normal physiology are very important. So you can see how proteins interact with other proteins. You can see how proteins interact with lipids, DNA, how proteins may bind a particular drug, and so forth. Oh, I think I have protein there twice. That's a mistake. Sorry about that. Okay, now when you run a 2G gel, so in the first dimension, you're just going to take this and actually run it in a column based on isoelectric points. So you only have one sample. And on a normal 1D gel, you have many samples that you run in one dimension. So here you start out with one sample, okay, You run it based on isoelectric point with isoelectric focusing. I don't even know what IPG is. Don't ask me. I'm sorry. I got this slide off the internet and I didn't find out what IPG was. So, but it takes about 17 to 72 hours to run isoelectric focusing. And then you get this band pattern, and then you take this band pattern and you put it at the top of a gel, and then you just run it based like just doing SDS page, and you separate out based on molecular weight. And then when you've done that, each one of these blots or dots corresponds to a particular pure protein, okay? And so I can cut any of these out, digest them, and then run them with a mass spectrometer. And then once I've run them with a mass spectrometer, then identify what protein that is within the sample. So you can get many proteins. Now, this is a very simple gel. If you really run two-dimensional gel electrophoresis, you're getting thousands and thousands of proteins, so it would be much more complex. You can also look at capturing target proteins to look at protein detection. And I don't even like this slide. I don't like the way this is target proteins that bind other proteins, and it doesn't do a good job. I'm going to skip this. There are other ways that we can talk about how antibodies can be used to detect protein binding. It's basically the principle of a protein chip, and you just have a detector that will detect when two proteins interact. And I don't even like the way this is set up, so please ignore that slide. And that happens to be the last slide. And I think what I will do, let's go ahead and look. Rather than starting chapter 5, I am going to wrap up this video lecture right now, and we will have a short video lecture to finish up chapter 4, and I will see you in class on Monday.